You don't just live in a plastic world. Plastic is living in you, and your brain is not exempt. For decades, we believed the brain was protected. The blood-brain barrier, a biological firewall, selective, controlled, stable. Heavy metals, blocked, pathogens, stopped. Large foreign particles, rejected. It felt secure. But something synthetic has now been measured inside human brain tissue. Plastic. Plastic was engineered for durability. That durability is now the problem. Microplastics, particles smaller than 5 millimeters. Nanoplastics, small enough to approach molecular scale. They are in oceans, in soil, in rainwater, in seafood, in bottled water, in human blood, and now documented in human brains. In 2024, researchers at the University of New Mexico analyzed postmortem human tissue samples. They detected measurable concentrations of microplastic particles embedded in brain tissue. Not surface contamination, not lab artifact. Embedded fragments. The dominant polymer? Polyethylene, the same material used in plastic bags and food packaging. Here is the anomaly. The brain is supposed to be shielded. Plastic is not supposed to cross that barrier, but it did. The blood-brain barrier is not a concrete wall. It's a living filter. Imagine bricks that can open and close microscopic gates. Glucose passes, oxygen passes. Most foreign particles do not. But nanoplastics exploit three vulnerabilities. First, transcellular transport. Cells engulf tiny particles based on size, not intent. If a particle is small enough, it may be internalized. Second, the Trojan horse effect. Nanoplastics bind to proteins in the bloodstream. Wrapped in biological material, they appear less foreign. They cross as molecular stowaways. Third, barrier weakening. Chronic inflammation from pollution, metabolic disease, stress can loosen tight junctions. Permeability increases. Access expands. In animal studies, polystyrene nanoplastics have been detected in brain tissue within hours of ingestion. Hours. This is not a rare, once-in-a-lifetime event. This is permeability under modern exposure conditions. And exposure is constant. The 2024 analysis revealed something more concerning. Brain tissue showed higher concentrations of microplastics than liver or kidney samples from the same individuals. Stop. Think about what this implies. The brain may not just be exposed, it may retain. In some samples, concentrations reach thousands of micrograms per gram of tissue. This is not symbolic contamination. This is measurable physical presence. Even more striking, individuals diagnosed with dementia showed significantly higher microplastic concentrations compared to neurologically normal controls. Correlation is not causation. But the pattern exists. Why might the brain accumulate more than detox organs? The liver filters, the kidney excretes, the brain stores. Once inside neural tissue, clearance is limited. Microglia, the brain's immune cells, can engulf debris. But they did not evolve to process synthetic polymers. If removal fails, particles persist. Persistence means chronic immune activation. Chronic activation means inflammation. And neuroinflammation is already implicated in cognitive decline. Engagement, reset, stop again. Here's the real question. Is escape from this plastic saturation even possible? Or are we only negotiating dosage now? Write one sentence in the comments. Escape is possible, or only reduction. Commit to one. At macro scale, plastic appears inert. At nanoscale, it behaves differently. Nanoplastics have enormous surface area relative to their mass. That surface binds heavy metals, persistent organic pollutants, plastic additives such as phthalates and bisphenols. The particle becomes a chemical carrier. If lodged in neural tissue, it may slowly release adsorbed compounds. Rodent studies have shown increased oxidative stress markers, reduced synaptic protein expression, impaired spatial learning. Oxidative stress damages membranes. Neurons depend on membrane precision for signaling. This is not acute toxicity. It's structural interference. Imagine pouring microscopic sand into a supercomputer. The machine doesn't explode. It glitches. How does plastic enter the bloodstream in the first place? 3 primary routes. Inhalation. Indoor air contains microfibers shed from synthetic textiles and furniture. Ingestion, seafood, salt, produce, bottled water, drinking water. Even treated municipal systems can contain measurable particles. Estimates suggest humans ingest tens of thousands of microplastic particles per year. Possibly more, depending on lifestyle. Nanoplastics can cross the intestinal lining. The gut barrier shares structural similarities with the blood-brain barrier. If one is permeable, the other may be vulnerable. You are not exposed occasionally. You are exposed daily. Now pause. This is not about panic. It's about dose. Elimination may be unrealistic. Reduction is not. 3 practical shifts. 1. Reduce plastic bottle use, especially with heat. Use glass or stainless steel when possible. 2. Limit microfiber shedding indoors. Synthetic clothing is a major source. Favor natural fibers when practical. Wash synthetics at lower agitation. Use microfiber filters if available. 3. Control indoor dust. Microplastics accumulate in household dust. Wet cleaning and HEPA filtration reduce airborne particles. These are not dramatic interventions, but long-term exposure is cumulative. Small reductions compound. And this is not just individual willpower, it is infrastructure. Water systems, waste systems, manufacturing systems. Most exposure does not feel like a choice. Here is the unavoidable conclusion. Plastic is no longer just an environmental pollutant. It is becoming a biological presence. 2 possibilities exist. Option 1. Microplastic accumulation is largely inert, a measurable but clinically insignificant artifact of industrial civilization. Option 2. We are witnessing the early phase of a chronic, population-level neurological modifier. A slow variable, difficult to isolate, easy to underestimate. Long-term longitudinal data does not yet exist. Causation has not been established, but presence has. Particles once thought too large to enter have crossed. Barriers once thought secure are permeable. Stop. Think about what this implies. Plastic production has increased exponentially for 70 years. Human neurobiology evolved without it. Now the integration is measurable. Your brain is not isolated from your environment. It is continuous with your civilization. The question is no longer whether plastic is inside us. It is what persistent synthetic matter does to a biological system over decades. Measurement is new. Accumulation is not. And once something crosses the barrier, the definition of outside changes permanently.